Now, the third case study, the microbial discovery. So in this case study, a scientist is interested to improve their understanding of coronavirus of their interest using the whole genome sequence data from NCBI within a secured environment. We choose coronavirus as a subject because it's a hot topic and everyone wants to know how we can use this case study to understand better, but you can use any other model organisms. But as a quick note, for this case study, we are using data from NCBI and not from any of our clients. Here is the project plan. So once the scientists sequence a coronavirus of interest, for example, MT334553, the next question comes in their mind if this genomic sequence is a novel or already known. If it is unknown genomic sequences, that means they discovered a novel virus. On the other hand, if it is a known genomic sequence, then it is not a novel virus. But either way, they'll be interested to know the genomic differences and their evolutionary relationship with other SARS-CoV-2 viruses. So now you got the sequence and very excited to know more about these genomes. Now to answer the questions, what next, we need some suitable tools and database so that can answer our questions. So for that purpose, Loci can help in that. Simply click on blast menu on the navigation bar after logging into the Loci. And here, the blast results will help to discover if this virus is already known or novel. Now come to the blast page and paste the new SARS-CoV-2 sequence of interest and blast against the in-house pre-formatted blast database of 928 SARS-CoV-2 genomes, which we created on Loci, taking the data from NCBI. So this is a in-house pre-formatted database. It is not present other places, but we take the data from NCBI and create it ourselves from the Loci. Now these blast results will help the scientists to discover if the coronavirus of interest is a novel or known one. So here is the results of homology search of one SARS-CoV-2 sequence and with in-house pre-formatted SARS-CoV-2 database through the blast. And since we got the blast hit, so it is not a novel genomic sequence. Now we can discard the no part and more focus on the yes part. And then next we study the genomic differences and similarities with other SARS-CoV-2 viruses. For that, we did the multiple alignment of all these 928 whole genomes. And then we search where there are differences and similarities. We are particularly interested to find out the SNPs, and we found at least in 10 genomic regions that showed the variations when whole genome sequencing data of 928 SARS-CoV-2 sequence were compared. So if you see the consensus positions in the screen, so that indicate that there is the mutations and SNPs. And by digging deeper with the MT334553, when compared with the reference sequences of SARS-CoV-2, they differ only in 5 genomic regions, which are shown here. So if you look into the first figure, you will see that the consensus region, which is indicated by a tall bar, is actually absent on that particular location 8826 on the first figure. And here the nucleotide base changes from C to T, and that happened for other positions as well. So now the next step and the last step is to build a phylogenetic tree from the whole genome sequence data to determine the evolutionary relationship of MT334553 with other coronaviruses. So on the left, this is the complete phylogenetic tree of 928 SARS-CoV-2 whole genome sequences that were collected from the NCBI. And since the whole tree is very big, we keep it to fit in the screen, we keep it as a small one, and then we zoomed out the regions where this MT334553 are located. So it is indicated by this pink region. So that's where this particular genome of interest is located. And so if you see deeply, you will see that the phylogenetic tree indicates MT334553 is closely related with the MT259266 from USA Washington and MT308694 from USA Michigan. So currently, the multiple sequence alignment and the phylogenetic tree features are in developmental phase and not yet integrated on the loci, but still we wanted to share these features in this preliminary form. So in conclusions of this case study 3, scientists are able to discover the similarity, variations, and evolutionary relationship of SARS-CoV-2 of interest using loci. Now they can conduct similar studies for other microbes or organisms of interest